So we have done the initiation of transcription. Now the next step will be the elongation. In this process, RNA will be formed. For understanding elongation, let us look at the DNA structure once again. Here are the two strands of DNA. One is template strand. Template strand is also known as antisense codon. And the other strand is coding strand. Now the RNA will be formed from the DNA template. Does anyone remember which enzyme was used the most in this process? It was RNA polymerase. RNA polymerase II specifically. This is because mostly messenger RNA is made by the process of transcription. Now, what will this RNA polymerase II will do? Well, it performs several functions. First, these two DNA strands will be separated from each other. Who will do that? It will be done by RNA polymerase. Then this separation should remain fixed. Now this stabilizing will also be done by RNA polymerase. Then, this DNA needs to be read and will be converted into RNA strand. Let's say this is the strand from 3' to 5'. So the RNA polymerase reads from 3' to 5'. And then will synthesize starting from 5' to 3'. So RNA polymerase will perform 5 functions. It will open the DNA. It will stabilize the DNA. It will unwind the DNA. Then it will read the DNA. And lastly, it will synthesize RNA from DNA template. Remember the functions of RNA polymerase biomnemonic. SOURS. S for stabilize. O for open. U for unwind. R for read. S for synthesize. An important drug is rifampicin acts on RNA polymerase and inhibits its synthesis in prokaryotic cells. So, that is why the process of transcription stops when you take rifampicin. And this drug is used in the treatment of several infections. This is important for your USMLE exam. So do remember that. We said that the DNA is from 3' to 5' or from 5' to 3'. So, what does it mean? Let us tell you, as we like to keep you familiar with the details. If we look at the structures of nucleotides, then this is the phosphate group. And this is ribose sugar. And here is the nucleotide. Now phosphate group is the 5' or 5' end. And the OH portion is the 3' or 3' end. Then let us start the process of reading the DNA template. If this is the guanine in DNA template, the other base that would be made opposite to that will be cytosine. If this is the cytosine in DNA template, The other base that would be made opposite to that will be guanine. If this is the thymine in DNA template, the other base that would be made opposite to that will be adenine. If this is the adenine in DNA template, the other base that would be made opposite to that will be thymine? No. It will be uracil, as there are RNA strands. So, that is how the RNA will be made from the DNA template.
Explore our extensive library of over 1,800 video lectures to learn about a wide range of topics. Only on sqadia.com.